Welcome, everyone, and thank you for joining us this morning or afternoon to this webinar. Before we get started, I wanted to point out two things. First of all, we are recording this webinar, and we will send you the link to the recording during the next days. On the right-hand side, you see a Q&A window. Please use this window and type your question in there. Only myself can see the question, and I will read out your question to today's speaker in the Q&A session. I forgot to mention who I am, sorry. My name is Carla Bellack, and I'm the marketing manager here at Mountain Thermal Systems. And I'm now going to present you today's speaker. It's Dr. Frank Elson. He's the Senior Application Scientist for the PatchClamp systems. He worked as a research scientist at Seattle Children's Research Institute before joining MCS in 2012. Today, he will give a presentation of about 30 minutes, and afterwards, as said before, he's available for the Q&A session. So I will now hand over to Frank.
Thank you, Carla. Good morning, everybody, or good afternoon, wherever you are. Like Carla said, please feel free to use the question and answer chat box, and we will get to the questions after the talk. And with that, I think we get started. Okay. Carla, is it possible to see the mouse?
Yes.
Yeah, so good. Yeah, that's the title is, the talk is about the pipette-based automatic patch clamp machine that we have, the PatchServer. A quick overview over the outline of the talk. Just to bring everybody up to the same speed, I will quickly talk about the automatic chip-based systems and compare them to the manual patch approach so that everybody knows how the chip-based automatic systems work, which is different than how the PatchServer works, which is an automatic system that is on the pipette-based approach. Okay. So the automatic systems, like some of you might know, they are based on a planar approach. You have a chamber where cells are floating in, and the machines then create a negative pressure, which sucks the machine onto the trapping channel, and which then creates the cell-attached patch configuration. But most of those machines, they work actually in a different configuration. They go into whole cell, and this is the standard mode of those machines. This is the basic technical approach of those chip-based machines. As some of you might know, with the manual patch clamp, it's different. The manual patch clamp needs to use pipettes. And here we have a pipette that is being moved towards the cell. So not the cell is being moved, the target, the pipette is being moved with a positive pressure. And when the cell is being approached, then this positive pressure is being switched to a negative pressure. And then we'll see how the seal is being formed. This can then also lead to the whole cell procedure or to the whole cell configuration. And there are two more configurations available in the manual patch approach. One is the inside-out configuration, and the other one is the outside-out configuration. So there are four configurations that you can record of with the manual patch approach. This manual patch approach is basically what the PatchServer uses. So important to note here for the PatchServer, I want to make that clear. It is not a standalone acquisition system like you might be used to our MEA systems that you purchase and then you put them up and you start recording. The PatchServer is more like an add-on system that you add to an already existing manual patch clamp setup, or of course we can build the manual patch clamp setup from scratch and then add the PatchServer to it. It will make the patching basically simpler and more efficient. Technically, I would say we can call the PatchServer an automatic patch clamp system that utilizes manual technology. Here you see an overview of our test setup that we have here at MCS. Important is, of course, that there are acquisition amplifiers, either with HICA EPC10 or with the multi-clamp 700B. Either one will work. And then there's the main unit, which is the heart of the whole system. And then on the stage, there are several components, and I will go now, will introduce you to each component now, one by one. If you look a little closer, you see how these components are aligned on the stage. On the left-hand side here, you have the cell reservoir and the catch pipette, as well as the ultra-fast application tool. And on the right-hand side, you have the recording pipettes. In this case, it's a 4-channel recording pipette system. And this is a close-up view of how the pipettes are arranged, how that looks. These components are connected with each other, basically with three different ways. There are data control cable connections. Then there are pressure tube connections between the components and mechanical connections. And I will put this overview on the side so that you can now, while we introduce the components, you can always go back and forth and see how they are actually connected with each other. The first main part, and that's why we also call it the main unit, is a component that is the central element, and it contains the pressure controllers, which is a roll pump together with the sensor, and it coordinates basically the entire patch process. Then there is the catch pipette, and the catch pipette is basically this little glass pipette here. Yeah, it sits inside a holder, and that holder is connected with a pressure tube to the main unit. So the main unit can control the pressure inside the catch pipette. In addition to that, inside this holder here is an amplifier that is also of importance, and I will get to that in a minute. And you can also adjust basically the position of the catch pipette with a manipulation of the holder. Then there's the cell reservoir. The cell reservoir is basically this part here. Yeah, it has a lid to it that can be opened up, and the cells are being filled into this little container. Usually 200 microliters is sufficient. The main purpose of the cell reservoir is to host the cells and to deliver them to the catch pipette when needed. And here's another close-up of the cell reservoir. And you can see the capillary which guides the cell close to the catch pipette. Then there's another part on this cell reservoir, that's the dispersal device. It's basically this syringe, and we use it to create a little steering effect inside the cell reservoir container. And this tube here is connected to this dispersal, this pressure tube here is connected to this dispersal tube here. So by moving the syringe a little bit back and forth, you can gently steer the cells when they started to settle down inside the container. Then there's the ultra-fast application tool, which is an optional component. And it is being used to investigate fast desensitizing ion channels. And I will get to this tool specifically in a separate slide. Then there are, of course, the recording pipettes, and important to note here is that you can use standard class capillaries that you have to pull in your lab by yourself. We do not sell any pre-manufactured recording pipettes. As all manual patchers know, you have usually you have a puller in the lab and you create your own recording pipettes. Manual patchers call them usually patch pipettes. Yeah, so you make them yourself. And important to note is that the 4-channel system, you can disconnect the pipette holder clip, and you can fill the pipettes and mount them into the clip, and then the clip is easily attached to the array. The advantage here is if you have a second clip, it can be already prepared while the first clip is being used for recordings. Then there is another option where you have a 1-channel system with 1 recording pipette. The bath chamber is also slightly different to normal bath chambers. To accommodate a 4-channel system, the bath chamber itself is a little elongated. And important to note here with the bath chambers are these 4 application bays. In the 4-channel system, we have 4 recording pipettes. And during the experiment, the recording pipettes are actually being moved into into separate application bays. And important to notice here that each application bay has its individual inline. So you actually can do 4 separate experiments at the same time. So basically the PatchServer patches a cell and serves it to the user ready for the experiment. And this is done in 5 steps. The first step is that the recording pipette has to locate the tip of the catch pipette. And this is important because each recording pipette is of course slightly different in shape and size. Therefore, the physical position of the opening is always different between each recording pipette. Yeah. So the first step is, like I said, the recording pipette has to move forward and find the opening of the catch pipette, which I call the cell catch position, because that will be the position of the cell later on when it is being caught by the catch pipette. The next step would be that then cells are being released through the capillary into the cell catch zone. And the next step is that the catch pipette catches a cell. Now I have to point out that all these steps are fully automatic. No user interference is necessary. Then the next step would be that the recording pipette moves forward again and establishes the desired recording configuration. Again, fully automatic with direct feedback control. So this is now repeated for all 4 recording pipettes. And then each recording pipette will have a cell attached to them. And then the last step would be that the recording pipettes are being moved all together into the application base. And that's when the experiment starts. Okay, I think we need to move a movie. Let's see which one. It might be this one, I assume. The movie doesn't really run very well, and so we have to see. I think it should run like this, or color. So, okay, this is better. Yeah, thank you. So, okay, this is a movie, an animation movie about the process, how it works, how the PatchServer patches the cells. It basically explains the 5 steps again, just now in an animated way. Like I said, the cell reservoir is hosting the cells. The capillary guides the cell to the catch pipette. Then there's the catch pipette with its holder and the amplifier inside. This amplifier has nothing to do with the acquisition amplifier. I will explain in a minute what it actually does. Then on the other side, we have our recording pipettes with the holder. Then we have the bath chamber, like I mentioned before. Important to note here is the application base. The size of the application base roughly 30 cubic millimeters. Then the main flow can be switched off and on. So now here we have our main players. I stopped the movie for a second so that I can explain a little bit what happens now. In a minute you will see how the whole process goes forward. The catch pipette now, like I said, is the place where the cell, when they fall out, being caught. But before that, like I said, each recording pipette, the tip, has to find the opening of the catch pipette. This is done electrically. And here comes the amplifier of the catch pipette into place. The amplifier of the catch pipette sends out a signal that is being recorded by the recording pipette, and then the manipulator is being moved to move the recording pipette towards the strongest part of that signal, which is at the tip of the opening. That's basically how it's done. And we can see this in a minute in the movie now. Moving closer, so the signal is being sent out. The recording pipette records the signal and tries to find the strongest point, basically the origin of the signal. And as soon as that happened, the recording pipette is being moved back, and then the cells are being released automatically from the cell reservoir. And now the amplifier of the catch pipette has a second function. It now measures basically the resistance of the catch pipette. So as soon as a cell is being caught, the machine knows that the cell is in place. And here's an important point. The machine then gives you the option, you, the user, has the option to stop and look at the cell. If the cell is to your liking, if you think it is worthwhile to be recorded, then you just go ahead and let it be automatically patched. If you don't like the cell, you have the option to reject it. The cell is being blown away and a new cell is being caught. So this is an important point. If you have very expensive cells, you do not want to waste your resources and time on cells that are actually not worthwhile being recorded. So the cell is being caught. That is being seen by the cell, by the amplifier. The recording pipette comes forward, establishes the seal and the recording configuration that you need. Then the second pipette does the same thing. Has to find first the opening again, pulls back, cell is being caught, and recording configuration is being established. Then comes number 3, number 4. We skip this finding of the opening. It has to do that, of course, too. And when all 4 have a cell attached to them, then they are being moved all together into the application bay. And now the user takes over and does the experiments. Okay, yeah, Carla's helping me here. Thank you, Carla. We have another movie that shows actually the process in real time. Yeah. So we just started and we let it run. So we have the catch pipette, we have the capillary. The capillary sits usually on top of the catch pipette. This is how it is. And here you see already how the recording pipette tries to find the tip of the catch pipette. And this is about how fast it goes. And please note, in this example, we even had some dirt inside the recording pipette, which, as a manual patcher, you might know, is a bad thing. The PatchServer doesn't really care. It works even with dirt in the recording pipette. Now we see the cells are being released, and if you don't like the cell, you just reject it. It's being blown away. And if you don't like, if debris is caught or something, you don't like it, it's blown away. If you do like the cell, you basically approve, you say okay, and then the cell is being patched. And this is done under direct feedback control, which means the resistance, the seal resistance, is constantly monitored. And the machine applies the pressure as needed, the suction, basically negative pressure as needed. Now you can see how the membrane is being sucked in. and then when the seal is being established, after that, in this case, it goes into wholesale, also fully automatic, and again under direct feedback control, and establishes the recording configuration. And when that happened, then the catch pipette will let go. While this whole process is happening, the catch pipette holds the cell with slight negative pressure. And when it lets go, it basically switches to slight positive pressure to let go of the cell. Okay, so these were the movies that we wanted to show you. And that's also, I think, almost at the end of our presentation. We still have a couple of things. So let's go on. So like I said before, we have the ultra-fast application tool. Most of you might be already familiar with it. Just briefly, you use these kind of tools to investigate fast desensitizing ion channels. And what you have is a Theta class pipette. It is a pipette that has that creates two distinct laminar solution streams. And you need a fast piezo, a step motor basically, with a controller that moves the pipette sideways. And how does it work? Well, you can imagine we are looking here now at the tip of the pipette, and there will be a constant stream of solution, control and test all the time, coming out of the tip. Then you take your open recording pipette, no cell attached to it, and you place it at a certain position, and this is the position that you have to determine first. You place it inside the control solution. Then what you do is you move the tip, and not you, the piezo moves it. And please note that the tip of the pipette is being moved, not the recording pipette. And this is done super fast. So usually we have, if we find the position very nicely where the recording pipette sits, then we can have a very nice and clean onset and no disturbance afterwards. And this position that guarantees it, we call the recording location. Here comes now the very good point with the PatchServer. The PatchServer, as you know, has a catch pipette, and you can mount the UFA tool with its Theta pipette above the first application bay, so that they are physically being mounted like this to each other. Now, the first step, as I said before, is for the recording pipette to find the cell catch position. Then you move the recording pipette over, and this has to be done now manually the first time, and you find the recording location. After you found the recording location, you can save that location to the computer. Now the computer can basically calculate the two positions, how they are located to each other. And from now on, if you go and get a cell, get a whole cell, then you can automatically let the computer move the cell into the recording position, and you can start your experiments. And this is being possible because the cell catch position in blue and the recording position in red are two fixed positions in space. And if you know one, you can move to the other one automatically, which will of course speed up the whole process very nicely. So I will have this summary sheet here that quickly connects the features of the PatchServer to its related advantages. This fully automatic patch clamp procedure has several advantages, of course. You can establish all the known recording configurations of the manual patch clamp. It is very simple to use, easy to handle. You can use the entire range of patch clamp protocols. Any patch clamp protocol that you can do on a manual patch clamp setup, you actually, in theory, can also do with the PatchServer, because the PatchServer is being mounted to a manual patch clamp setup. The data quality is guaranteed, always the highest, the gold standard. Giga-ohm seals and the plugin into the different recording configurations is always done with direct feedback control, so that guarantees that all the recordings have the same quality. And another point here that I didn't mention so far is that while the recording configuration is being established, all the vital recording parameters are being monitored and logged, which gives you, of course, a good control afterwards to see how good was my recording configuration actually. Then, like I said, we have a 4-channel system, which of course will increase the data production rate. And we use standard glass pipettes. We use only few cells. The rest of the cells can be basically sucked back into the cell reservoir. So we do not lose many cells, not as many as you would normally expect. And then during the experiment, we do those experiments inside the application base, which guarantees you a very small area, and therefore you need only very little test solution. And all these points, all these features basically guarantee you low operating costs. Then this feature is really important, I think. You can visually identify the cell before you go into patch it, or before you let the machine patch it. And this, of course, is very valuable if you have expensive cells. You could imagine that, for example, if you have a certain channel being expressed in your neurons, then you can put a fluorescent tag to those channels, and you can see if a cell expresses well, it should be bright in fluorescence. If not, then not. And doing that, you will have a nice measure of the cell. Is it worthwhile your resources and your time? Then another feature of the PatchServer is that it is a modular construction, which will help us, of course, to basically give you only the parts that you really need. So if you're already in the possession of an EPC10 HEKA amplifier, you of course do not need to buy a new one. You can just use the one that you have. And then there is the ultra-fast application tool. For some of you, this might be a very interesting part if you are interested in fast desensitizing ion channels. Okay, possible applications. From the microelectrode array, you know that our microelectrode array has many applications. The PatchServer applications has a few less. So we can do cell cultures. We can also do acute slices, but only if you take the cells of the slice and bring them into solution. Cardiomyocytes, stem cells, and so on. And we already did recordings on several of those cells, which I will quickly show you in this slide. All experiments were done at the NMI, either with 1 or 3 channel prototypes. And for example, if these are experiments done on alpha-1 nicotinic acetylcholine receptors, and if you look at these recordings, you cannot really tell which is which. In this case, the manual is on the left and PatchServer is on the right. And in general, the PatchServer recordings had very similar quality, I think even better quality than when you compare them with manual recordings. Then the following data slides are all done with the PatchServer. Here we, for example, have CHO cells, alpha-7 and alpha-1 recordings. And this is done with the prototype of the UFA tool. You can do, for example, desensitization recordings. And when you overlap them, you can see the longer you wait with the UFA tool for the second pulse, the more the channel recover. These are typical experiments that can be done. Then we do have an example here of a 3-channel prototype that was a prototype for the multi-channel recordings that we had. Initially, we had a 3-channel that we now increased to 4-channel. But these recordings were done with a 3-channel, so you can do 3 experiments at the same time. So, for example, dose response curves can be done much faster. Then, of course, cardiomyocyte. Here's a recording that shows a beating cardiomyocyte that we recorded for. So this is just a few examples. This brings me to the last slide, the summary slide. And like I said, the PatchServer automatically establishes the recording configurations, and it employs, to do this, it employs step by step, basically the manual patch approach. And important to note also, to remember, is the low operating costs. And the PatchServer itself is a modular constructed add-on tool. So please remember, you cannot just buy the components and then start recording. You need actually manual patch clamp setup to do those recordings. And that brings me to the end of the talk. And if there are any questions, please don't hold back. Just write them in the answer in the box, and we try to answer them. Thank you.
Yeah, thank you, Frank, for this presentation.
Yeah, thank you. Can I go back? I think sometimes, Carla, it's
sure.
It's nicer if we do 1 second. Okay, Carla, go ahead. So yeah, it's nicer this way. Then
to have the overview.
Yeah, exactly. Thank you.
Yeah, so actually there was a question whether the PatchServer provides a fully unattended recording procedure. You were talking a lot about automatic recordings and whether it has to be attended or not. Yes. So basically the idea of the PatchServer, first of all, is of course that it provides you with the cells in the desired recording configuration fully automatically. And then the idea is that the user, if wanted to, can take over and do the experiments themselves. So that is the basic idea. Now, it is possible, of course, for example, with the EPC10 and the PatchMaster software from Hika, you can follow up the patching of the cell with the automatic experiment. So I have to say, yes, that is possible. You could, if you want to, design the entire experiment ahead of time, and then let the computer basically, or let the PatchServer patch the cell. After it's being patched, move it into the application bay, and then have the experiment being started automatically from the PatchMaster software. So yes, it's possible. Another question was whether the PatchServer can establish giga-ohm seal formations and provide high-quality recording configurations sufficient enough to measure small ionic currents. Yes, absolutely. So basically, the giga-ohm seal formation is, of course, the first step, as you all know, and in that formation, the cell-attached patch formation, you can, of course, record single channels. Yes, and it does this, like I explained before, with direct feedback control, I call it. So it continuously, while it establishes the seal, for example, looks at the seal resistance. And you know, as an experienced patcher, sometimes you go and to seal a cell and it seals within seconds. Sometimes you have to apply a little bit more pressure, a little bit less, and you have to fight a little bit for it to get the seal. And the same is, of course, true for the PatchServer. But here, the good thing is it actually can respond to the individual approach. And therefore, it can establish always the same quality of giga-ohm seal. And it does the same, basically, when it goes into whole cell. It continuously looks how good is my configuration. And if it's not good enough, then it will try to improve it until it has a good recording configuration. Okay, thank you. Mm-hmm. Another question was about the data points per day one could get. So do you have any estimate on how many data points? Yeah, that's that's super good question, because that's, of course, the whole idea. Like all manual patcher know, the success rate, the automatic patch clamp field likes to call it, the success rate in manual patching depends a lot on the condition of the pipette, on the condition of the solutions being used, on the condition of the cells being used. All these points are really important, and because the PatchServer uses the manual approach, these points are also important for the PatchServer.
So I have to say, if all those conditions are in a good state, in an acceptable state, then the patch server will patch every cell that is being provided. And I would say it's above 90% that the patch server is able to patch a cell in a high quality recording configuration. Yeah, we do have the whole idea of 4-channel system is now that you can do those 4 recordings at the same time. So if you patch a cell, and let's say you can get a cell within 2 minutes. Yeah, so within 8 to 10 minutes, you will have 4 cells being patched by the 4-channel system, and then you can do a 5-minute experiment. So roughly, it is basically that you would get maybe 4 cells being recorded every 15 minutes, something like that. Yeah, and then that would calculate with, if you have an 8-hour day, that you will get roughly about 100 data points per day. So when I say data points, it's basically 100 experiments per day, which is a lot for manual quality recordings.
Okay.
Good.
Thank you.
Thank you very much.
I think this was it for questions.
Mm-hmm.
So thank you for joining us today. As promised before, we recorded this webinar and we will send you the link to the recording. Please note that we have some upcoming webinars on the 2 electrode voltage clamp systems. We're talking about the high clamp, our system for small compound volumes for Xenopus oocytes on Thursday, and next week we will have the webinar on the RoboCyte 2.
Cool.
Yeah, if you have any question, you see our phone number and email address below. Please just give us a call or drop us an email.
Yeah, and anytime emails to me if you have any questions, no problem.
Yeah. When I send around the recording, I will CC Frank so you have his email address.
Super.
Yeah. We will stay online for another 5 minutes or so in case any question comes up.
Sure.
And yeah, thanks again for joining. Thank you, Frank, for the presentation.
My pleasure.
Have a nice day.
See ya.
Bye-bye.